privilege to be here. My name is Steve Byard. I work for Arcinova. Title of my presentation is Optimizing Drug Substance with Expedited Product Translation to the Clinic. There's a QR code there. That is basically a checklist if anyone wants to engage in the kind of information that we would like prior to sort of helping you to move forward. Then that QR code will give you some guidance on that. It's an absolute pleasure to be here today, simply because we're relaunching our name Arcinova. And I'll say a little bit more about that in a minute, but we are based in the UK, in the north of the UK. It's a very large facility. There's a picture there. That's basically, imagine that as 2 football pitches if you want some idea of the scale, so it's a reasonably sized facility. The important thing is, over the last few years, we've invested tremendously. We've invested $7 million in our chemistry facility. But the bottom line, in terms of our core services, what we do is we translate molecules from that sort of point of discovery through to first in human and beyond if required. So we're covering all the disciplines, and I will go into that in a minute, but basically the chemistry, the analytical, the formulation development, the manufacture, some of the regulatory, quality assurance, etc., that is all taken care of. Just by way of introduction for myself, my name is Steve Byard. I'm a PhD organic chemist. I don't want to go into this into too much detail, but you'll see there that basically I've published quite a lot. I have strong links with academia, including a professorship at Warwick, and also I stand on the Parliamentary and Scientific Committee at Westminster more recently. Also I sit on the Board of Directors for North East Process Industry Cluster. That's quite significant. The bottom line here, what I'm trying to convey, the message is that we are a science-led organisation, but also we recognise business as well. So that interface of where science meets business is quite important for us, and we make sure that we do actually tailor the science towards your business needs. If we drill down a little bit there in terms of the core capabilities, the core disciplines that we have on site, we do take care of the drug substance, lots of facility, lots of capability here in terms of knowledge, intellectual power, but also the hardware associated with drug substance manufacture, straight from PR&D through to larger scale manufacture. It's all there. Isotope labeling, that is a unique service offering. Often it's 14C, carbon 14, but we do stable isotopes as well. Drug product, obviously once we've made the drug substance, we need to translate that to the clinic. So again, we take care of all the formulation development, straight through to manufacture under GMP conditions, and that is Annex 1 as well for parenterals. Finally, once the drugs have been in the clinic, phase 1 often, sometimes phase 2, then we can take the plasmas, the bloods, etc., the urines, and do the bioanalysis to support the mass balance studies. It goes without saying, underpinning that, we have solid-state analytical sciences. That is a unique service offering in its own right, but equally we have microbiology on site to support the drug product manufacture. And as you would expect, quality is the lifeblood of our organisation, so we have quality assurance on site in conjunction with regulatory affairs. So I'm just going to start to drill down into this a little bit, because we're all involved in drug substance manufacture, and really one of the unique offerings that we offer is that relationship between studying at the molecular level the structure and dynamics with respect to bulk properties. So really the bottom line here is understand your molecule. Make sure you're minimizing those unknown unknowns. And again, we have the tools to do that. So here you can see there's an orthogonal set of axes, that relationship between structure and dynamics with respect to properties, but also the orthogonal axis of processing with respect to performance. The ideal situation is that people who are working on the chemistry, i.e. understanding the molecule, the manufacture, understanding the final finished product, understanding how that relates to the final performance. And if you get all that under one roof, then that really sets you up for really, I'd say, a seamless transition of getting that molecule into the clinic. It goes without saying that we have the instrumentation, we have capabilities to do that. We have equipment such as high field NMR, we have 500 megahertz NMR there, and I'll say a little bit more about that in a minute. But equally, we have all the supporting equipment, all the separation sciences, all the physical solid state sciences to support all this. I've not made mention of this thus far, but we do have, for example, formulation or manufacturing equipment such as arc melt extrusion, spray dry dispersion, and it goes without saying that we can actually support all the formulation development to underpin that. But the bottom line here is what I call pharmaceutical engineering, and really when you're setting out on that journey of developing that molecule to get it into the clinic, really there are lots of design space. It's an extensive design space which we have to operate, in which we have to operate. This, for me, is an opportunity and not a challenge. You can actually shape the entire trajectory of that drug by getting it right at that point. If you want to get it right, understand things at the molecular level, really probe that molecule at the molecular level, understand the dynamics, understand the structure, and understand how that's going to work with respect to any excipients bearing in mind your target product profile. So get it right there, understand the molecule, and that basically the drug ability will follow with some reasonable ease. Again, this molecule is important to you. It's unique. You're developing it simply because it has unique properties. So to apply a generic process to that, it's not something that we consider really appropriate. So we do actually partner with you. We work in a bespoke way right from the onset. So the minute you come to us for a proposal, we really get the NDA, CDA in place, and really look at what your targets are, what your business model is, and what your technical requirements are, and make sure that we're dedicated to your exact needs in getting that molecule to first in human. A lot of people often forget that the dossiers are required for registration. Okay, that's not on people's mind at the beginning, but equally, if you get this right, you will actually facilitate that process. So the bottom line is get the drug substance right, and a lot of other things will follow. I make no apology for sort of launching into some science at this point, okay, just to show you a little bit about the sort of things that we are capable of doing. Again, that's a DSC, that's looking at thermal analysis of a particular drug substance. And you'll see there's a low temperature endothermic event. The question is, what is that? Is it a polymorph? Is it not a polymorph? What are you going to do about it? Sometimes it's there in some drug substance batches, in others it's not. It may actually be sort of considered the next step is to go to XRPD to look at that in a bit more detail, variable temperature XRPD. And what you'll see there actually is over that transition, the X-ray pad diffractograms are roughly the same anyway. Some slight differences in relative intensities, but that's not really telling you anything about that material. The bottom line here, again, is a case of how do you probe at the molecular level? The conventional techniques don't always get to where you need. You need to remove that unknown, remove those uncertainties. And one of the things that we can do is probe at the molecular level using solid-state NMR spectroscopy. So these are the C13, CP mass spectra, solid-state NMR spectra over that transition. And without going into too much detail, you can see significant differences between those spectra. The bottom line is below the transition, 2 molecules per asymmetric unit, above it, 1 molecule per asymmetric unit, and below it, if you come back, you show that that's reversible. I should also say that 3 weeks ago, actually, I was invited to open the national facility of the 1.2 gigahertz NMR instrument in the UK. I have direct access to that as a professor of that facility. And again, that's giving us that extra leverage in terms of understanding your molecule, taking care of those unknowns, probing at the molecular level, and making sure that we're getting that seamless transition from the chemistry through to the clinic. I've mentioned the fact that we do formulation development. That is also non-clinical as well as clinical. This is a recent example. Often you'll find for oral solid dosage forms, people suspend in Methocel or carboxymethyl cellulose. This is something we did recently, and you can see straight away under the microscope, this is a simple technique, but under the microscope, very, very different morphologies. And really that's a red flag, because actually what's going on there is that molecule is hydrating. That will be a stable hydrate. That means it will be less soluble, and by implication you will have less bioavailability. Really what I'm saying is, as an organization, we can take care of that under one roof and make sure that we're considering at the molecular level, straight through the bulk properties, with a vision or with a line of sight on how we're going to get that through non-clinical into a clinical environment. I'm also showing, loosely related to that particular drug actually, some SEM images, electron microscopy images on the right-hand side there. Those allegedly were different batches, but the same material produced by the same process. But actually, if you look closely, the upper image shows some very small network-like fibrils, which are actually changing the properties of that drug substance immensely. I can tell you now the surface area of that particular batch was 50% greater than the batch below it. That clearly has implications in terms of suspendability, in terms of dissolution characteristics, and by implication in terms of bioavailability. The question is, understand the chemistry. How are you going to control that? Is it a critical quality attribute? What are you going to do about it in the chemistry lab? And equally, understand the downstream properties in terms of the translation to the clinic. Similar thing, again, I don't make any apologies for delving into science here because it is the core of what we do. And again, just to be mindful of the fact we're interfacing science with business, so we're not trying to create second PhDs or anything like that, but we really are trying to close that space, minimize those unknown unknowns. Here, again, X-ray powder diffraction, diffractogram shown, different peaks appearing. The question is, what are those peaks? Again, delving down at the molecular level, you need to go to the molecular level to see this. Solid-state NMR comes to the rescue, and you can see straight away which nuclei are actually giving rise to those differences. A lot of you may be familiar with two-dimensional spectra for solution state. We can now do two-dimensional spectra for solid state and really make sure that we are pulling these molecules apart, metaphorically speaking, to understand what's going on there. Finally, when you've got through this, you've got the drug substance, you fully understand it. How do you translate that to the clinic? We are Arcinova. Our parent company is Quotient Sciences. We do engage with something known as translational pharmaceutics. That gives us the adaptive approach towards basically formulating at the clinic, removing some of the need for some of the stability studies, really accelerating that molecule into first in human, giving ourselves the option to actually change the formulation, change the dose at the clinic. And again, that's proven in terms of saving money and saving time. Subsequent to clinical studies, again, I made mention of the fact that we do take urines, we take samples, and any metabolites that we discover above 10%, then obviously we have to characterize those. We have published on this quite extensively. A lot of what we do is confidential, but here's a publication that's gone out. That's the work that we did. That was a major metabolite of aclabrutinib, which is now on the market. But again, you can see that publication, see some of the work that we did in terms of characterizing that metabolite. I do have limited time. Okay, I've taken up a little bit more than I had right to do, but all I can say is thank you for your attention. We do have a stand, 93. Liz Dawson and A1 Chong, both business developments based in the US, are present as well as myself. Again, there's a QR code there. That is checklist that you may want to fill in if you have any interest in coming to work with us. Time for a few questions if there are any. Otherwise, so a question at the back there, yeah. We did, okay, and thank you for asking that question because it's a case of what is a polymorph and what isn't a polymorph, actually. Sometimes that endotherm was there, sometimes it wasn't. We did resolve that. I can't say too much on that, but the solid-state NMR showed us exactly what it was, which part of the molecule was responsible for that, and also that informed us, first of all, how to control that from the chemistry perspective, but it also informed us what the impact would be in terms of bioavailability should that molecule be used in a solid oral dosage form. So that's a really good question. I'm more than happy to talk to you about that later, actually. But a lot of people do regard polymorphs as discrete thermodynamic entities, and sometimes that's not the case. You get some more of a continuum. Sometimes you get physical forms that are stabilized by the presence of another physical form, so they coexist. Okay, they're not just discrete domains. Okay, so I can talk to you about that if you're interested. Yeah. Okay then, yeah. Okay, so there's a question there. So the question there, it's about the design of the drug substance in terms of, I think in terms of optimizing bioavailability through dissolution. Okay, and one thing that I didn't mention is the fact that we do salt selection, we do polymorph screen, also physical form screen, but you can actually tailor the drug substance both chemically, in terms of counter ions, in terms of physical form, but also do the pharmaceutical engineering. You can do size reduction in various different forms or modify using different additives as well. Okay, but that is something that we do specialize in, and again, that's about understanding the drug substance at the molecular level and what excipients you may wish to use with that. Okay, and sometimes, in some cases, we actually take those excipients as part of the synthesis as well. Okay, and you can actually use different counter ions or co-crystals to help in that dissolution process or bioavailability process. I do have limited time, but I'd like to follow up with you on that question actually. But it's a really good question. Okay. Okay, thank you. Yeah. Yeah, we took offline maybe. Exactly. Yeah, and that's actually what buys us the advantages of getting from drug substance to clinic efficiently. You're absolutely right. Yeah, exactly. Yeah. Okay, so I think my time is up now, actually. But again, thank you very much for your attention. It's been good to meet you thus far, and I really do hope you get time to pop by and see us on our stand 93. Thank you very much, everyone.